Hey everyone, I want to show you the genetic code and its error correcting feature. Very interesting feature. A new paper just came out that refers to this error correction feature as a possible way of evolving proteins. Protein evolution is a problem for evolution. There's really no way that proteins are going to evolve by random chance. And so evolutionists have always been trying to figure out how to solve this problem. And now they've hit upon this idea of maybe the very code itself, the genetic code, could have within it some secrets, some special capabilities that help to produce and evolve proteins. Let's have a look. First, let's have a look at this paper. There it is right there. Here's the passage that talks about the genetic code and how it is has this kind of built-in safeguard for error correction. So let's have a look at this. First, for some quick review, here is just a sequence of nucleotides that I made up. This would be an mRNA sequence for messenger RNA. That's when the DNA a copy is made of the DNA, and that copy is called messenger RNA. It's going to be edited and then sent to the ribosome to be translated. I did an earlier video on all of this, but just basically each one of these letters is a nucleotide. A for adenine, U for uracil, C for cytosine. So each letter is just an abbreviation for a small molecule called a nucleotide. These nucleotides are going to be interpreted 3 at a time. Those triplets are called codons, and so that's the genetic code. You take 3 at a time, and that is going to be translated into a amino acid, in this case isoleucine. So AUC, adenine, uracil, cytosine, is interpreted by the ribosome as isoleucine. It'll end up grabbing an isoleucine from the environment and gluing that on to this chain of amino acids that it's building. The next one in this case, CGA, would be arginine, and then leucine, and so forth. So just an example there. Now, what if it got shifted by 1? This happens sometimes with what's called frame shift mutations. You have a mutation in the sequence, maybe an insertion or a deletion of a nucleotide, and it could cause these codons to be shifted. Could be shifted by 1, could be shifted by 2. You can also read these things backwards, which we'll look at in a minute. So in this case, now you're not reading AUC as your first one, you're reading UCC. And that gives you a completely different amino acid. So if you have this frame shift, you end up getting a completely different sequence of amino acids. In this case, there's even a stop signal, UGA. So let's have a look now at the genetic code and how these assignments came about. So here is the typical way of representing the genetic code itself. So you have these triplets, or codons. In the upper left-hand corner there you have the first one, uracil. Uracil codes for phenylalanine. UUC also codes for phenylalanine, and so forth. So you've got these all these triplets. There's 64 of them in total. Some of them code for stop. That tells the machinery, okay, this is the end of the protein. And then you've got 20 of the typical amino acids that are used in your cells, in all of our cells, to construct proteins. So these are the 20 amino acids. There's some interesting things about this code that jump right out at you. When this was discovered more than 50 years ago, and they should have seen these non-random patterns right off the bat, and it should make you ask what's going on. So here's the most obvious one. I'm outlining in red the oily amino acids. So what do I mean by oily amino acids? So here's a chart of the 20 amino acids organized by chemical type. So these top ones, these green ones, are the oily ones, and I'm outlining, taking a look here at isoleucine. Let's home in on that. Here is a blow up of isoleucine. The green part is the side chain that sticks out and gives it the amino acid its chemical properties. In this case, you'll see it's all carbons and hydrogens. It's one hydrocarbon, and that is oily. That's like olive oil or something. It doesn't mix with water, it's oily. So those amino acids were the oily ones. So back to our chart, most of the oily amino acids are in the first column. So And these are the ones that are used the most often also. So really about 90% of the oily amino acids that appear in proteins are in the first column of the genetic code, the way that it's depicted in this table. Obviously, this is not by chance. Obviously, this is not random. Obviously, there is a structure here. Something is going on. This is interesting, right? So they should have asked this question right off the bat. Something is going on. Now, we still don't know all the answers, and there's a lot of other features of this code I won't talk about in this video. I'm just hitting on this one just to give you a flavor of the kinds of things that the genetic code has to offer. What does it mean? What's the impact, or at least one of the impacts, of having a column of all the same chemical property, in this case, oily ones? Well, if you were to read it backwards, it would you stay within the column, basically. Let's just have a look at an example of this CUU here that codes for leucine. If you were to read that backwards, it would be UUC, which takes you up here to phenylalanine. If you're going backwards, if you just think about it, that means that the middle nucleotide doesn't change. You're swapping the first and the third of these codons. So when you go backwards, the middle one is conserved. Well, that's what a column is in this table. A column is defined by keeping the middle one the same. So you're just flipping around within the column when you go backwards. Yes, you're changing positions, but within the column. So any amino acid that is oily that appears in the first column, if you go backwards, it's also going to be an oily one. Let's have a look at back to our example here. So we looked at this example here, and I just made this one up, but you can see that 4 of these happen to be oily: the isoleucine, leucine, another isoleucine, and valine. Let's go back to our table just to show you that. So there's the valine in the lower left, VAL. Then you have your isoleucine above that, and then you have your leucines. So these are very strongly hydrophobic. And now we're going to go backwards. I'm going to show you what that looks like. If you go backwards, you get rid of those first row, and now they switch. So the isoleucine turns into a leucine. The leucine turns into a valine. This isoleucine turns into a leucine, and this valine turns into a methionine. Hydrophobic for hydrophobic. Really interesting. You have a structure, a non-random structure that means something's going on. This is a designed code. This isn't just a random code that jumped out of nowhere. It is not random, and in fact, as the more we learn about it, the more we find out that it is incredibly sophisticated. The genetic code is really something, and we still haven't figured out all the secrets. I'll talk more about the genetic code in coming videos. Until then, religion drives science, and it matters.